My name is David Hardy. I've been at UCLA for a while, although not currently employed here, but we get the opportunity to introduce our speaker for today. And first of all, I want to just extend a debt of gratitude to Helen Holdsworth, who is responsible for reaching out to Romas Geleziunas to bring him here today. So thank you very much, Helen, for making this happen. Our speaker today is someone who I think is very unique and sort of, I think, an unearthed treasure for me that I didn't really know he had been in the area of HIV work as long as he has for the past 28 years, which is pretty, I think, remarkable. Dr. Romas Geleziunas is a director in clinical virology department at Gilead Sciences. He received his PhD in the laboratory of Mark Weinberg at McGill University in Montreal, and did his postdoctoral studies at University of California, San Francisco with Warner Green. As I mentioned, he's worked in HIV for the past 28 years, both in academia as well as in the pharmaceutical industry, and he's authored over 70 publications. I got the opportunity to go online and look at a lot of those today, and he's published some very notable both depth as well as width in terms of this area, working not only with antiretrovirals, but immunomodulators, and most recently in the area of HIV cure. He has established and has led Gilead's HIV eradication program. He is currently the head of the HIV Advanced Therapeutics Group at Gilead, and also a member of the NIAID ACTG HIV Reservoirs and Viral Eradication Transformative Sciences with TSG. Prior to joining Gilead, he also worked in a number of drug discovery programs at DuPont Pharmaceuticals, later acquired by Bristol Myers Squibb, and also Merck Research Laboratories. So without further information, please invite Dr. Geleziunas up to talk about strategies to achieve antiretroviral-free HIV remission, industry perspective.
Thank you. Thank you, David, for that very kind introduction. I need to tell you that the last time that I spoke at UCLA was in 1992. I was interviewing for a postdoc position in Irv Chen's lab, my idol, and still is my idol. I had a nice chat with him this morning. It was terrific. So I'm glad to be back many years later, and today I'm going to take you through, I think, some very interesting stories. As you can see from the title, we're thinking way beyond antiretroviral therapy. We're thinking of eradicating HIV infection. And as you can imagine, this is a very complex journey, and I'll take you through this. There's some really interesting things to discuss. Okay, so I think this audience understands very well that antiretroviral therapy doesn't cure HIV infection. In fact, when a patient presents, they suppress with antiretroviral therapy their plasma viral loads very well. Of course, if they stop therapy, in the vast majority of cases, there are exceptions, we know this, they basically rebound quite quickly. So the key to this field is, what is the cause? What is the source of the rebound, even after decades of antiretroviral therapy? And the source is believed to be a series of reservoirs. Some of them we understand better than others. Others are more complicated. The one that we understand the best is this memory CD4 positive T cell. And the reason we understand it the best is because everybody who works in this field is able to readily isolate these cells from the periphery of patients, either through large blood draws or leukapheresis, and we're able to study them. So for me, in my business, in industry, it's critical that we have the ability to isolate them and test concepts. There are other reservoirs that the field is starting to talk about, but those are harder to isolate, hence it's hard for us to test interventions. So the reservoir is at least one reservoir that we are concerned with is this memory T cell reservoir. Very infrequent, occurs at around 1 per million cells, but its half-life, importantly, is very, very long, 44 months, and this is why it's hard to eradicate this reservoir. So we and most of the field have been looking at this problem in two ways. One, as I said, this particular reservoir happens to be latent most of the time, so if you want to basically deal with it, you have to expose it first and foremost. And by exposing it, I mean you need to find a safe modality to activate the expression of HIV transcripts as well as proteins. And in this regard, what we in the field have studied are HDAC inhibitors. These are inhibitors of histone deacetylases. I'll tell you quite a bit about this. We've learned quite a bit about these agents. PKC agonist, that stands for protein kinase C. Quite a bit of work has been done in the field on this as well. We have done HTS, high throughput screening. We've done a lot of that. Nothing much has come out of that. It's been a frustrating journey. But what we have done is we've, I think, identified a new way to activate latent AIDS viruses, and that's through the use of a toll-like receptor 7 agonist. I'll tell you that story. We've just recently gone public with that, so that's a very intriguing story. Now, once the cells are exposed and they start to express viral proteins, you can start imagining kill strategies. You can start imagining ways to basically target these cells with, for example, therapeutic vaccines. I'll tell you about my favorite one in the field. The use of antibodies that target GP120, I'll tell you about that as well. Immune checkpoint inhibitors are very popular right now in this field for the same reasons as cancer. And again, I'll show you the new data that we have. The toll-like receptor 7 agonists may also have a role in reducing these particular reservoirs. All right. So this question, can current in vitro assays and methods that we have help select LRAs, that stands for latency reversal agents, that's what we call activators in this business, for clinical testing? This is a very important question that we have obsessed with quite a bit, because being an industry, our issue is we need to translate things to the clinic. And obviously, with the numerous concepts that are on the table, it's not possible to throw everything forward into the clinic, so you have to have methods that basically enable you to reduce the risk or increase your probability of success in being able to identify agents that are worth moving forward. So what we have done here, and I'll take you through the thinking, is we've borrowed from the many years of experience that we have at developing small molecule antiretrovirals. And this is a very interesting story because, in essence, in the early days, it wasn't quite clear how to select these agents. But over time, not just Gilead, but everybody in our industry has developed a series of very simple procedures to identify agents that are worthwhile moving forward. And that is very simply identifying or determining the EC50, the inhibitory concentration, the EC50 for HIV in a simple cell line. This is very, very easy. The other component is understanding protein binding. So you have to really understand the free fraction, the fraction of drug that is not bound by plasma proteins. This proved to be critical in selecting agents to move forward. And finally, some animal PK. So with an EC50 against HIV, protein binding to plasma, and some animal PK, we are really good at identifying the right agents to move forward into clinical testing. Of course, we don't have that for the latency reversal, for the HIV cure space right now. That's what we're trying to define, these criteria that are going to be useful to move compounds forward. So, and what I'm going to tell you about next few slides, this is only applying to LRAs, latency reversal agents. The various other classes that I spoke about in the previous slide, antibodies, vaccines, immune modulators, we have no idea of how to select those. We don't have any metrics by which to try to be able to move these forward into the clinic. So let me take you through how we reasoned and how we basically arrived at pushing things forward into clinical testing. So here, and this is many, many years of research that basically is summarized here. So the way we look at agents is we want to basically make sure that the agent is active in primary latency assays or cell lines, but that is not sufficient. That, we have learned over the years, is not sufficient. And so what we do is we have to, we run these particular agents that you'll see in patient cells. And we find that this is an absolutely critical piece. And this is obviously cells from either leukopaks or blood, and we basically measure the expression of HIV. Now, what the field has done, we look at, and this is because, as I said earlier, these reservoirs are very infrequent. They occur at a frequency of 1 in a million cells. So you have to use very sensitive PCR-based methods to basically measure this cell-associated RNA. But we've also discovered that that is not sufficient. So what we do is we take it a step further, and what we basically look at is the induction of viral RNA in culture supernatant. And this is basically an indication that actual viral particles are being made. And you'll see why that's important in a few seconds. All right, the other thing that we have learned is, and when you have the opportunity, is to make sure that the LRA, the latency reversal agent's concentrations that are achieved in vivo are basically at or exceeding the concentration that is required to activate the virus in vitro. And you'll see why that's important as well. So I think this set of criteria have been useful for us in selecting agents to move forward into clinical testing. So let me tell you about the inhibitors of histone deacetylases. This is the area that has received the greatest amount of attention, and so let me take you through this carefully. So histone deacetylases are enzymes that are in the business of removing these chemical moieties called acetyl groups, and specifically acetyls found on the amino acid lysine. In terms of substrates, these enzymes work on histones, tubulin, and transcription factors. And what the result is of the action of these enzymes is suppression of gene expression, including HIV expression. And there is really very good agreement in the field that everybody who has looked at these HDAC inhibitors in latently infected cells, the vast majority of investigators have seen these to activate expression of HIV. So in vitro, we were in very good agreement. Now, this class of drug enabled us to leap forward to the clinic and basically ask, okay, of all these agents and the criteria and the measurements that we're doing, do any of these predict any type of efficacy in the clinic? All right? So that's what we're going to look at in a few slides. But before I do, I just want to quickly tell you about the mechanistic basis for this. So if a gene needs to be activated, what it does is it basically recruits to its promoter histone acetyl transferases, HATs, the opposite of an HDAC basically. And these HATs basically, as you can see on this figure, add these acetyl groups depicted in green, and what this does is it loosens up the chromatin, enabling now transcription factors in RNA polymerase II to access the gene and initiate transcription. Okay? Now, the opposite or the opposing effect is governed by these histone deacetylases, and what they do is they remove these acetyl groups, pushing the chromatin back in a condensed condition where, again, you're in a repressive state. So, bottom line, you can use inhibitors of these enzymes, these HDACs, to temporarily push the situation into one that favors transcription. Okay? So that's the simple principle behind the mechanism of these agents. Now, as I said, there are agents that are FDA approved in this area, not for HIV, of course, but for CTCL and multiple myeloma. And so this enabled a series of groups around the world to test whether these HDAC inhibitors would do something to perturb reservoirs, to induce expression of reservoirs of HIV. So the players in this space are vorinostat, a drug manufactured by Merck, panobinostat, recently approved by the FDA, manufactured by Novartis, and romidepsin, a drug that is manufactured by Celgene. The structures are at the top, again indicated for various cancers, and the second line is basically the various groups that studied this. So the take-home message to us is this, that these compounds vary in their potency. This is the EC50 to induce HIV. As you can see, SAHA is significantly less potent than either panobinostat or romidepsin is to induce HIV. And I want to leave you with this message as well. So this is a very key metric, I think. So in the case of SAHA, if you look at the Cmax, the maximal plasma concentration of this drug, that concentration corresponds to about 10% of what is needed to activate HIV in vitro. So you see what we're trying to do. We're trying to connect in vitro to in vivo. Of course, this agent doesn't look so great. But in the case of panobinostat and romidepsin, you have, in their plasma Cmax, you have a 2-fold excess or 11-fold excess over what is needed to activate latent HIV. So this would be favorable, okay? So now let's start to look at some of the data that kind of led to the proposal of the testing of these agents in the clinic. So these are the 3 drugs that I just told you about, romidepsin, SAHA, and panobinostat. And what we did here is we've tested these drugs in specifically memory CD4 T cells from ART-suppressed subjects. And what we do, which is a bit different than the rest of the field, we expose these cells to the duration and concentrations of drug that are achieved clinically. So we don't hammer them. We don't put a 10,000-fold excess and leave the drug on for 5 weeks. We don't do that. That does not mimic the clinical situation. Okay, so in this particular case, we basically used a 40 nanomolar concentration of romidepsin. These are the concentrations of panobinostat, and we basically treated them for either 4 hours or 24 hours in the case of these 2 drugs. And then importantly, we carefully monitored over time the expression of HIV. And what you see is that the drugs are not identical. They're from the same class, they hit the same types of enzymes, but they're not quite identical. So in the case of romidepsin, you see the expression of HIV come up very quickly at 6 hours, and this expression persists for 24 hours. The other 2 drugs are more transient in nature. You see it comes up at 6, down by 48. And this nicely correlates with the action of these drugs in cells. So this is looking at histone or HDAC activity in the cells. And as you can see, the drug romidepsin has a persistent effect on this enzyme, whereas these are transient, showing that there is a correlation which suggests to us that there's a mechanistic link, that the inhibition of these enzymes are somehow governing the expression of HIV in patient cells. So this is where the field stands. We run these types of compounds in a variety of assays. Pretty much everybody agrees that this happens. But the thing that started to bug us is this is RNA expression in cells. It doesn't mean that these drugs are inducing viral protein expression, and that's central. Remember my first slide, I said the goal of this strategy is to expose virally infected cells. We want these cells to basically start to produce proteins or minimally viral peptides so that the immune system or various strategies can see these cells and then destroy them. Okay? So it's really important that we induce viral protein synthesis. And as I said earlier, that's a very hard thing to do. These are rare infected cells. The only way you see HIV in them is by using sensitive PCR-based methods to look at RNA. All right? So the way we kind of approached this question was by looking at supernatant. So cells are making the viral RNA, but the question was, could we find this viral RNA in the supernatants? And if we could, this would be an indication that perhaps virions are basically taking off or egressing from these cells and then ending up in the supernatant. So here's, again, the two drugs that I've been talking about, romidepsin and SAHA. And now this is, again, conditions that mimic the clinic, very importantly. And what we're looking at here is the effect of 5 nanomolar romidepsin versus 40, looking at culture supernatants versus SAHA, again, at conditions achieved clinically. There's a very clear difference here that obviously everybody sees in the room. One drug is inducing the egress of I think, particles and RNA appearance in the supernatant, and the other drug is not doing that. And further, what we have done is we've taken these soups, and again, these are just tricks to try to convince ourselves that these are viruses. We take these soups, and then we basically pellet them at high speed. If these RNA molecules pellet, it suggests that they're in larger bodies, proteinaceous bodies, likely virions. Okay? So this is how we basically convinced ourselves that one drug might be better than the other one to move to the clinic and induce viral protein expression. Now, this slide is just basically a quick review of how we helped the two clinical studies that tested romidepsin in the clinic, how we helped them select the drug concentrations. So what we did here is we basically estimated the concentrations, 3.5, 15, and 40 nanomolar, that are achieved by dosing 0.5, 2, and 5 milligram per meter square of this drug. And this is the actual CTCL dose, or the dose given to cancer patients, which is 14 milligrams per meter square. As you can see, for HIV patients, we're proposing a fraction of the cancer dose. And so these are the doses that are being tested in an ACTG trial called A5315 and in a Danish study called REDUC that I'll tell you about in a few minutes. All right, so now let's go look and see what happened in the clinic. Now, I kind of set the stage for you. I'm telling you that SAHA doesn't look so great to us based on the metrics that we use. Romidepsin looks like it's somewhat more promising. What happens clinically? So this is a very nice study, a pioneering study that was done by Dave Margolis and published in Nature a few years back, and he tested SAHA, and this was basically 8 patients that he looked at, and he found that the drug vorinostat, and these patients, I have to say, were pre-selected. He basically tested these patient cells in vitro to make sure that the drug worked in them before he actually dosed the same patients. Okay, so trying to assure himself of success here. So the same 8 patients, and as you can see, baselines are in white, black is vorinostat or SAHA. In each of these patients, there was a nice approximate 5-fold average induction of cell-associated RNA. Very importantly, they didn't see anything in plasma. Okay, so this is, again, a very important distinction. You can induce transcription of HIV in cells ex vivo. You seem to be able to do this with a single dose in vivo, but you're not seeing plasma RNA. Now, the other thing that happened with this particular drug is when they started to dose this on a daily basis, that fold induction decreased to around 2-fold or barely activatable. So there's obviously complex mechanistic issues that are going on here. So take-home message, these drugs seem to translate from the laboratory to the clinic. SAHA seems to induce cell-associated RNA in the lab as well as in the clinic. Now let's take a look at the other drug. So this is the REDUC trial that was conducted in Denmark. And the investigators there basically tested 3 doses of romidepsin given once a week. This is the first dose, second dose, and third dose. And now you're looking at plasma viral RNA copies. So a very different story emerges here. There seems to be a drug-related induction of viral nucleic acids. This is using the COBAS assay, so it measures nucleic acids in plasma. Now, it's very interesting that the first dose didn't do very much. The second dose was clearly better in 5 of the 6 patients, and the third dose, only 1 patient responded. So again, mechanistically, very, very odd what's going on, very complex. Don't quite know what the reason for this is, but there seems to be, in this particular case, a drug-related effect on plasma. So we think that this might be related to what we had seen in vitro, which is the induction of viruses in culture supernatants. So, bottom line or take-home message on the HDACs and what we have learned over the years, and this is what we're going to be doing moving forward. We find that these HDAC inhibitors, they're not all identical. They mechanistically work the same, but they're not identical. And what we feel is important is to make sure that you move compounds forward into clinical testing that have Cmaxes or plasma concentrations that are equal or in excess of what is required to activate HIV in vitro. Again, borrowing from the antiretroviral days where we were basically ensuring ourselves that in the case of antiretrovirals, it was actually Cmins that were important. We would make sure that at Cmin, the concentration of drug is in excess of what is needed to inhibit. So this concept is really the same. It's at Cmax asking whether the drug is in excess of what you need to hit the target. And then the other point is that ex vivo HIV activation, what we do is the drug needs to be able to show an effect on culture supernatants. It's not sufficient to show the effect in cell-associated RNA. And so far, we have very limited amount of data. Very few trials have been conducted in this space. But the trend that we are seeing from this limited set of data is that it's possible that these types of metrics may help in figuring out what is worthwhile testing clinically, because romidepsin seems to have done something that SAHA has not done so far in the clinic. So that's essentially what we have done for the last many years, and we've basically set the stage and feeling a little bit more comfortable in what we're advancing into clinical testing. Now, what I'd like to show you next, the next series of slides, is this really very exciting series of studies that we have done that has led to some really intriguing data that I think the field has received very positively. So I want to take you through that. So this is the Killer7 story, and the program actually started 10 years ago at Gilead. And this program was initially launched because at the time we were looking for a replacement for pegylated interferon for the treatment of hepatitis C virus. We felt that if we could have a way to have the patient induce their own interferon locally in the liver, that we have better efficacy and fewer side effects compared to interferon, pegylated interferon, which is administered systemically. All right, so that was the initial rationale. But as you'll see, this drug has had a very interesting life, and it has been tested in HBV and HIV, and will be tested in HIV indications as well. So Killer7 agonist, specifically 9620, is a clinical candidate at Gilead. And what it does is it engages the toll-like receptor 7, which is expressed in dendritic cells and B cells. And when it engages this receptor, it induces a signal transduction pathway which culminates in the production of interferons and pro-inflammatory cytokines. This agent is very selective compared to others that preceded it. And when tested in models of hepatitis B virus, we found that it reduced surface antigen in woodchucks and in chimpanzees, and in some woodchucks induced seroconversion. So profound biological activity with a single small molecule agent. And so this is what basically motivated the company to move into HBV-infected patients in clinical studies, and that's where we are at right now in phase 2 studies. Now, importantly, what we're trying to do with this drug is to stay away from the induction of peripheral interferon. We're trying to basically reach doses that simply induce interferon-stimulated genes to, again, stay away from the side effects. Okay, so now I'm going to tell you about the HIV story and how we basically arrived at thinking about this agent in the kick and kill strategies that I talked about early in the presentation. So in terms of kick, what we have found over the many years that we've studied this agent is that 9620 and many other analogs of Killer7 agonists induce HIV RNA in PBMCs from HIV-positive subjects. We've also seen this agent, this series of agents, activate CD4 T cells, but it's a small fraction of them. But that's where some of HIV resides, at least in certain reservoirs. And in terms of kill, we know from years of research, again, that these 7 agonists enhance antigen presentation by dendritic cells, just like the natural ligand, which is single-stranded RNA, does the same thing. And importantly, what we found is that these Killer7 agonists activate Cd8 T cells and NK cells. And these are cells that are in the business of lysing virally infected cells. So we like that attribute. Importantly, and I want you to remember this, these Killer7 as well as Killer8 agonists actually induce antibody-dependent cell-mediated cytotoxicity. So this is the phenomenon by which NK cells recognize virally infected cells that are coated with an antibody and via the action of perforin and granzyme B lyse the target cells. Okay, so remember that for later on. So here's some of the data. So we basically, just to assure ourselves that this agent worked well in inducing or activating CD8s and NKs from patient cells, we looked at cells from 6 patients, and we looked at CD69 activation of CD8 cells and NK cells, and as you can see in each of these patient cells, we had a nice response. So this is the effect that I was telling you about earlier. This effect was confirmed in phase 1 clinical studies, so it's not an in vitro effect, it is also an effect that was noted in clinical studies. Okay, so now let me take you through what was the key foundation for bringing this concept forward to clinical testing in HIV-infected subjects, and that is this series of data that was performed by my colleague Derek Sloan and his team at Gilead. And so what they did is they looked at the effect of Killer7 agonists in those same types of assays that I described earlier for you for the HDAC, so looking at supernatant viral RNA. So specifically, again, CART-suppressed patients, plasma viral loads below 50. In this particular case, we're using PBMCs, not CD4 cells, because the target of this drug is dendritic cells and B cells, but the receiving end is the CD4 T cell. Hence, you need to have a series of cells, the immune cells, that are present together to achieve this effect. Okay, so you need PBMCs here. And these are from leukapheresis, and basically testing a couple of different concentrations of the drug, looking at cytokines at day 2 as our pharmacodynamic marker to make sure the drug is on board. And then importantly, the RNA or the HIV bit is basically an analysis of culture supernatants by the COBOS PCR method. Okay, so let me take you through some data now. So what is evident in this slide is that this effect, this Killer7 effect on the production of HIV in supernatants is inconsistent. Right? You have patients here that respond to both concentrations of the drug. There are patients out here that are not responding to the drug. There are patients, like this one, responds at the low concentration. There are other patients that respond at the high concentration. This is a nightmare for somebody who works in industry. The effect is inconsistent, and it's not dose responding. Right? This is really, really bad. So this is why we really, really worked so hard for so long to try to understand this, and this is fact. This is not an artifact. This is what occurs in patient cells, and this is the limitations of experimenting in vitro. We don't quite know why this is happening, but remember I'll show you some data in a little bit, but there's a kick and a kill component to this class of compound, and it's possible that balance is what tips this effect into seeing the viral induction or not. So this was our reality, and the reality in this cohort of subjects is 72% of them responded by over 2-fold activation of virus at one of the 2 concentrations, and the mean fold activation about 9-fold. Okay, so that was reality.
For any given patient, is it consistent?
You mean longitudinally over time?
If you do the same patient.
Not even that. If you have a patient come back 6 months to a year later, those concentrations can flip-flop and some become less responsive. So it's very dynamic. The process is dynamic. Clearly, the immune system needs to be in a certain state to receive the signal from the drug and then communicate whatever it communicates to the virally infected cell. So that's what makes it really challenging. But I would say, hold a second, let me show you some in vivo data. The story changes a little bit. Okay, so we at this point said enough of the in vitro work, let's take a chance, let's go forward into an animal model of HIV infection, of AIDS infection, the SIV-infected Rhesus macaque. Let's see what happens in vivo, because we have a comprehension of this compound. It actually will work probably better in the context of lymphatic tissue, where all the cells are basically present in the right proportions, and it is lymphatic tissue. It's not peripheral blood, which is just one way of measuring viral blips. So what needed to happen, again, a large amount of work by a large number of people, dose-ranging studies in healthy cynos and Rhesus, and this was critical to establish what we call a minimal effective dose. And this is very important, because what you want to do is achieve the minimal dose that induces interferon, and also experiment with the idea of inducing just ISGs, stay away from peripheral interferon, and just have the response be an ISG response. This is also very, very critical. You cannot administer this agent on a daily basis. This will induce chronic inflammation. That would be a very bad thing. So this series of monkey studies were critical to establish the dosing frequency, and we now know from our work that once weekly or every 2 weeks is safe and tolerated in Rhesus monkeys. And the second piece that was needed was this. This is, everybody thinks that this happened overnight. It didn't. So you all well know that people have been treated with antiretroviral drugs, combination therapies for many years, but that had not been established for monkeys. And one reason is monkeys don't take tablets very well. They will spit them out. You will not get consistent effects. And if you crush these drugs, they have some bad taste and they won't use them. So monkeys are different than humans. To get a really good antiretroviral regimen for a monkey, you need to make it an injectable so that the PK is consistent, you can deliver it every day, and that's what we did here. So our terrific formulation folks at Gilead worked very hard and came up with a nice formulation that enables the assembly of tenofovir, FTC, dolutegravir in a single suspension that's injected once daily subcutaneously. All right. So now we have our in vitro data, we have these critical in vivo data, we have an ART regimen. It's time to do the proof of concept. It's time to go see what a Killer7 agonist might do in ART-suppressed monkeys. So here's the study. It's a collaboration with James Whitney at Beth Israel. And what we did, took 10 Indian Rhesus macaques, we infected them with MAC251 intrarectal, we gave them the subcutaneous ART at day 65, so at the beginning of chronic set point. We, at day 320 post-infection, that's 8.5 months of ART, initiated a dose escalation, not of 9620, but a very close analog of 9620. And this is dosed orally in the monkeys, 4 monkeys. We gave the agent every other week, and there were 6 placebos. The primary endpoint of the study was to look at plasma viral rebound kinetics after stopping ART, so the effect on reservoir. But there were secondary endpoints that were equally important to us, and that's looking at plasma viremia. The question is, what would this agent do in terms of plasma viremia? You remember now the romidepsin study that I told you about, the clinical study, small pilot study in humans, those blips were in the 20 to 100 copy range. Remember that, okay, as you go through this next series of slides with me. All right, so here's the CART suppression phase of the study. And as you can see, animals had very high levels of viremia. And here's when the CART initiated. As you can see, all animals suppressed. The animals that had higher set points took longer to suppress, as you can see, these guys over here. But eventually everybody does suppress, and the ART regimen is very solid, again, because the PK is so consistent given once daily. So after, as I said, 320 days, this is where we start the dose escalation of the TLR7 agonist. Okay? Now before I show you the virologic parameters, I want just to show you the effects, the PD effects. And I told you about the effect that this agent has on CD4 T cells. This is an indirect effect. It does not operate directly on CD4 cells. It is indirect, but here's the effect in vivo. So if you dose 0.1, 0.2, and 0.3 milligrams per kg of the TLR7 agonist, you see this low level but highly reproducible activation of a certain population of CD4 T cells. And then we basically stuck with 0.3 all across the board. Again, you're seeing this transient effect on CD4 activation. So good. Now this is the Cd8 cells. I told you these agents activate Cd8 cells. Well, here's the effect in vivo in Rhesus macaques. Again, the first 3 doses, seemingly a bit, maybe a trend here towards dose proportionality, and then it seems to taper off. But again, our Cd8 cells became activated as we had predicted they should. Okay. Now, let's go to the plasma viremia, and then this is what really we found to be very, very intriguing. So the first 3 doses, nothing happened. And as I keep saying to everybody, thank God we didn't stop the study here. We would have been really badly disappointed. Because the next dose, we started to see what is substantial blipping. This is about 500 copies per ml in 2 of the 4 animals. We give them another dose. Now 4 of the 4 animals start to blip. And these, again, these are substantial. This is 500 to 1,000 copies of viral RNA. This has never been seen in the field. Another dose, there it is again. Transient, beautifully inducible, comes down. The immune system clearly contains the blip, reduces these cells or the viruses to baseline again. And there it is again. So this was really blew our minds when we saw this, because to be able to induce with a pharmacologic intervention this kind of consistent transient blipping is exactly what we're hoping to do with this field, right? You have to expose these cells and then hope to have a second intervention that goes and kills them. So Conclusion is there is some sort of a priming event, which we don't understand. I look forward to anybody in the audience who has any thoughts on this. But initially, the first 3 doses didn't do anything. Then we get into this blipping phase. The placebo animals, none of them blipped. And in terms of the sequences, we actually sequenced this material. And this material actually corresponds to proviruses that are found in lymphatic tissue. And what we find very interesting and very promising, this is an important point, is that hypermutated sequences, we did see these sequences in viral DNA, in proviral DNA sequences, but none of these appeared in these blips. So, and I'm not a sequencing expert, so I can't really go into great depth here, but what the take-home message to me is that if this was a simple phenomenon of lysing cells and just basically disgorging viral nucleic acids, that hypermutated stuff would be present in the plasma, okay? But the fact that it's not hypermutated, this is clean stuff that's not mutated, it suggests to me that these are virions that are coming from somewhere in the body of the monkey. And so this, again, is, to me, a very positive sign at this point. Okay, I told you we designed this drug to induce peripheral interferon alpha, right? I told you that at the very beginning, 10 years ago, this program started by saying, can we find a substitute for pegylated interferon for the treatment of hepatitis C? So that's the primary pharmacodynamic marker for this drug, okay? So you should have on your minds, well, does it correlate? You know, does the blipping correlate with the goal or the mission of this drug? And the answer is no. These were the first 3 doses that I told you about. Remember I said that these first 3 doses did not induce any plasma viral RNA, yet they induce a very nice dose-linear induction of interferon alpha. So the drug is hitting the target, the dendritic cells. It is inducing interferon as it was designed to do. And then we have various animals at various time points responding to again, this drug and inducing interferon. So the bottom line is it doesn't seem to correlate very well with the primary pharmacodynamic effect of this drug. Now, what about viral DNA, right? So this is a reservoir measurement, not the most important one, because we know that a lot of the viral DNA is junk. It's mutated stuff that doesn't contribute to the viral rebound, but nonetheless, an easy marker of a viral reservoir. So here what we see is a very interesting trend to, this is after the 7 doses of TLR7, there's a trend towards lower viral DNA levels in PBMC, lymph node, and colon. Not all animals responded. As you can see, this one animal is not responding, doesn't see a decrease in viral DNA. But one thing's for sure, the placebo animals didn't do this. So an interesting trend that we are going to continue monitoring, and certainly, again, a positive sign that we've affected the reservoir in the right direction, i.e. a reduction in the reservoir. And the last piece is now bringing all of this together. So now I'm going to summarize for you what happened in this study, including the CART rebound phase, okay? So this is the front end of the study, viremic animals starting ART, suppression with ART. 320 days later, this is the blipping phenomenon in plasma. And here we stop the ART, let the animals rebound. Again, you see an intriguing pattern here, right? The animals basically in blue are the ones that receive the 7 agonist. The reds are the placebos. The kinetics of rebound didn't change, but the set points did change. There's a half-log reduction in the set point in the animals that receive TLR7 agonist. So again, a positive sign. What this means or what's doing this, we have no idea, but this certainly suggests that the immune system is better off now in containing this virus than it was before the drug was administered, because I'm assuming that this is the immune system that is doing somewhat better in these animals at containing a virus in plasma compared to these animals. So again, a positive sign. So, just to wrap this up for you, because again, I think this is an important development. We'll see if it's going to work in the clinic, but this is what we know so far from the 7 years of work with TLR7 agonists. They activate HIV expression in PBMCs from patients. They induce expression in not all, but about 2/3 to 3/4 of patient cells that we've looked at. They activate CD8s and NK cells. They show donor-dependent variation, and as I said, this is really something that bugs us a lot in vitro, and the best that we can come up with is that the kill part and the kick part are different in each patient over time. Now in vivo, TLR7 agonists I showed you induce plasma viremia in infected monkeys. We induce transient activation of CD4 cells, 8s and NKs, chemokines as well. We see a trend towards diminished DNA in these treated animals, and the viral set points seem to be lower after stopping ART. So as a result of all of this work, we decided to take the bold leap, and we said, let's go check this out in the clinic, because ultimately that's what matters to us. So we are currently enrolling a clinical study of 9620 in HIV-infected card-suppressed subjects. So we should have information by year's end and hopefully reportable by early 2016 on this. And so really it's all going to play out in the clinic as to whether this phenomenon translates or not. Okay. So I want to wrap up by now taking you in a different direction, which is now that we have this TLR7, or that you know that we have it, we've known we had it for a while, what does the 7 agonist combine well with? Now let's start to try to imagine what combination therapies might be beneficial in reducing reservoirs. So the things that we think about and that we think this 7 agonist might be compatible with is certain therapeutic vaccines. And the one that I'm most excited about is the vaccine that is being developed by our close colleague, Louis Picker. I'm sure many of you have heard about this story because it's quite fascinating. And so what Louis did is he basically took cytomegalovirus, Rhesus cytomegalovirus, and in it dropped SIV antigens and basically vaccinated animals and then challenged them with wild-type SIV MAC251. And he found something that no one else has seen in this field. And so let me take you through that. So this is vaccination of Rhesus monkeys with the DNA prime adeno type 5 boost vaccine. This is similar to the Vaccine Research Center studies and the Merck STEP trial, which used the AD5 as well. In monkeys, as in humans, this vaccine does not protect from acquisition of infection. So what Louis did, again, is CMV prime, CMV boost, so 2 shots of the same exact vector. He sees something different. So the animals so basically these animals are all vaccinated, and one challenge, there was 2 groups of animals, one that were not protected, but one group that was infected. And what's important about this vaccine is that it does not seem to block acquisition. As you can see, all the animals, this is plasma viremia, all the animals become infected, but half of them become controllers, the other half does not. Now what's very important, and this is through a series of very nice, elegant studies that Louis has published, is if he looks at these animals 3 years out, even though they were clearly infected early on, there is no virus to be found. So is this the blueprint for how to get rid of an AIDS virus infection? That's what's on our minds. That's what's on Louis's minds. The whole field is actually waiting for some data that I'll tell you about in a few seconds. So this is very promising to us. And what seems to govern the efficacy of this vaccine, at least in part, is the very wide breadth of immune responses that it's able to generate in contrast to other vaccines. So here what you're looking at is a series of monkeys, and you're looking at a gag response, CTL gag response in these monkeys. And as you can see, very many epitopes are recognized by the CMV vaccinated vaccines. This is an elite controller monkey, nowhere near what this vaccine does. And here are a series of other vaccines that have been in the field. Again, you don't see anywhere near this type of a response. So this wide breadth of a response could be what we need to basically contain an AIDS virus that's being reactivated. So, and I can summarize this really quickly for you. So this is many years of work for Louis, 50% protection in a prophylactic setting. We see control of SIV. There's an effector memory response that is robust, broad novel Cd8 responses, this intriguing phenomenon. There's some class 2 restricted responses. We won't get into that today. And eventually the control of SIV that leads to eradication. So the big question on everybody's minds is, well, that was very nice. It's a prophylactic setting. What would happen therapeutically? So we have a couple of studies that we're collaborating with Louis on, in which we basically infected Rhesus monkeys, gave them our CART regimen that I told you about today, the tenofovir, dolutegravir, FTC. We suppressed them, then we vaccinated these animals, and now we intend to release them from ART to see whether the vaccine in the therapeutic setting can do what it seemed to have done in the prophylactic setting. Okay, so this is a really important study for us, and so we're waiting for the results of this. It's a very nice collaboration with the Gates Foundation, Lewis's Institute, Jeff Lifson's Institute, the DARE Collaboratory, and Gilead. So this data should be available soon, and we look forward to seeing the results. So the other way that we have imagined that TLR7 agonists could be combined is with monoclonal antibodies. And remember, I told you to make note of the fact that TLR7 agonists augment ADCC, antibody-dependent cell-mediated cytotoxicity. So that's governed by these types of antibodies. So we feel that there's a good marriage there, that we might be able to have a nice kick agent with the TLR7 agonist, an enhancement of ADCC, which is mediated by antibody. And this clinically translates very nicely, as you can imagine, because we have a great deal of experience with antibodies. So why are we thinking of this? We're thinking of this because of this. So we can imagine that either with a latency reversal agent, i.e. TLR7, antigenic stimulation, or homeostatic proliferation, at some point in time, virally infected cells start to produce antigen, and specifically GP120, that goes to the surface of the cell. And some of these antibodies recognize and target these cells, and if we carefully enhance the Fc portion and arm it to be a very effective binder of Fc receptors, we might have the ability to recruit cells like NK cells, various phagocytes, or even get complement deposition to lyse these virally infected cells. And how did we become interested in this? It's because of the great work that has been done over the last 5 to 10 years by the Vaccine Research Center, Michelle Nussenswag at Rockefeller, and Dennis Burton and his colleagues at Theraclone and at the Scripps Institute. And they identified this new crop of broadly neutralizing antibodies that basically interact with various portions of GP120 or GP41. The one that I want to tell you about today is PGT121, which targets the V3 glycans of GP120. And we became very interested in these antibodies as a result of these studies performed by our colleague Dan Baruch in Rhesus monkeys. And so Dan basically took a group of viremic animals and infused PGT121, either alone or in combination, and made these findings. If the animals have a very high baseline viral load, the antibody is effective, transiently but very effectively suppresses plasma viral load. The animals do rebound. Intermediate levels, we see the same effect, a delay in rebound, but eventually these animals rebound. But this is the one that really caught our attention, these 3 animals over here. If you have a low viral load set point, you infuse this antibody, viral loads came down and stayed down for a very prolonged period of time. So of course, this led us to think, well, could it be that these antibodies got into lymphatic tissue and basically lysed, via the mechanisms that I told you about earlier, lysed these virally infected cells and specifically hitting key reservoirs, which is why this result led to a sustained period of viral remission. But even more importantly, you would ask yourself, all right, well, we can do this pharmacologically. What if we treated monkeys or people with antiretroviral therapy, bring down those viral loads to much lower levels, and then apply the antibody? What would happen then? So Dan did that experiment. It's the first iteration of this experiment. We're doing a different flavor of this that I'll tell you about in a second. But what Dan did in this next, this is again a monkey experiment, he basically treated the animals with our antiretroviral cocktail and started administering PGT121, the antibody, at the same time. Okay? And then he stopped the antiretrovirals. This is the antiretrovirals only without antibody, again, stopping antivirals. All of the animals rebounded that received the antiretrovirals, but in the small pilot study, 3 of the 5 were controllers, the ones that received the antibody. So again, consistent with this effect that this antibody is doing clearly more than the antiretroviral small molecules are, but what could it be doing? And this is where this extra piece of data was very informative to us. So the animals that became transient controllers, they did eventually rebound. They weren't cured, so this is not a cure for AIDS. Okay? So but they were transiently suppressed for many, many months before they rebounded. But what we noted that was very interesting, the animals that were controllers were the animals that had the lowest levels of viral DNA and PBMC and GALT. This does not happen in ART only. So the antibody is doing something different. It's clearly doing something different. It could be a variety of things. We like to think that maybe this antibody is going into tissues and lysing cells. Could also be limiting to transmission of virus and cell transmission in tissues. Maybe that combination of things is what results in this effect. But we do like to see this adjunctive therapy, basically with the antibody, doing something different that you see with antiretroviral therapy. Okay? So again, this was good enough for us to say it's time to translate to the clinic. Enough monkey studies, now it's time to go to the clinic. So what we did is we approached the company that owns the intellectual property on these antibodies, so Scripps, Theraclone, and we basically obtained an exclusive license from them, and our goal is to move something to the clinic as fast as we possibly can. The internal program that we have launched after acquiring these assets is to basically determine which effector function. I told you there's 3, there's ADCC, there's phagocytosis, complement. Which of these effector functions are best at basically reducing viral reservoirs? So we're going to try to figure that out. This is actively ongoing. And then to engineer these antibodies to basically contain these effector functions, and then, as I said, select an antibody for clinical testing. We hope to be in the clinic in about 18 months. Now, in parallel, what we're doing is we have, again, a very nice collaboration with Dan Baruch and the Gates Foundation This group, with our guidance and in collaboration with us, is basically advancing PGT121, the parent antibody that you saw in those monkeys. That's the exact same antibody. They're moving that into GMP manufacturing and are going to do a clinical study. So we hope to learn from these two studies which effector functions and what types of properties of the antibody make it more effective, if at all, at reducing viral reservoirs. So I think I'm going to wrap up and summarize for you what you heard here today. So I told you about our journey to try to understand how to basically bring small molecule agents to clinical testing. Try to select the ones that make the most sense. And through the years of research that we have done, we've identified certain criteria that we think are important. And out of this came the results that I showed you today. Killer7 agonists seem to be able to activate reservoirs or HIV and SIV in vitro and ex vivo and in vivo. So we're quite excited about that result. Hence, we're going to be focusing a lot of our attention on Killer7 agonists as kick agents. I told you that I think these Killer7 agonists are compatible with various other strategies that we are looking at that have in our portfolio or are collaborating with various people with. And that includes anti-envelope antibodies, PGT121. I explained why that is a compatible pairing, we think. I didn't tell you about bispecific antibodies. That'll be for another time. But I also told you about an exciting therapeutic vaccine. Remember, the Killer7 agonists activate Cd8 cells, and those are the cells that are elicited by the CMV vectored vaccine. So we think, again, kick and potentially enhancement of the vaccine effect. So we like that combination as well. The immune modulator, Killer7 agonist, as I said, enhances some of these effector cells. And obviously, the goal is to eliminate these reservoirs. We are imagining that all this would be done initially under the cover of antiretroviral therapy. So if we are activating reservoirs, we're not spreading viral infection. And that's kind of the scheme that we're following right now. Now, the last slide is, as I always say, the most important one because it acknowledges the important people that have done the work. The folks that came up with the nice monkey art cocktail is Bay and Jillian. The folks that did all the nice in vitro TLR7 is the team of Jeff Murray and Derek Sloan working under the guidance of Tomas. John Corbin is leading our broadly neutralizing effort, the optimization effort at Gilead. Tiffany and Joe worked very closely with James Whitney to do the monkey study of the TLR7 agonist. I told you about Dan and Dennis and Theraclone. That's where the antibody came from. We have a very nice collaboration with John Mellors at University of Pittsburgh on the romidepsin. We work with the ACTG and Celgene on this. And finally, and most importantly, we've had this very long collaboration, 5 years with Jay Lalazari and his tremendous patients that undergo the leukapheresis for us to basically do the work that you've seen, which is critical to advancing agents to the clinic. TLR7 romidepsin would not be possible, would not be in the clinic if it wasn't for these terrific donors that have given their cells to us for these studies that I've shown you today. I will stop here and take questions if you have any.
Do you see any analogy to the apparently resistant individuals who may have had a transient viremia, many of whom were also probably infected with CMV?
So all of the monkeys are CMV positive in these studies. This is really fascinating. So you don't have to be CMV negative. It makes life a bit easier for them. And often the monkeys in these colonies are generally all positive. So it's not that. It's not that the initial state of the CMV serostatus, if you will, does not seem to correlate with whether you become a responder to the vaccine, i.e. whether the vaccine enables the monkey to control the AIDS virus infection and that. So, and this is a question that Lewis gets every single time he talks. What's the correlate? You have a dichotomy. You have 50% responders, 50% don't respond. And to this day, he doesn't have an answer for that yet. But if he could crack that, that would potentially help improve the vaccine and hopefully maybe get those other groups, the non-responder group, to become a responder. But there's no indication of what is governing that yet. Back there, Judith.
That was a really terrific talk.
Thank you.
One question about the TLR7 pathway is, and this is not my area of expertise, but I think there's a sex link between TLR7 and responses. Have you looked at the sex of your patients or your cells to see whether there's a difference?
Yes, that's a great point. And also there are SNPs in TLR7, right? There are SNPs that are associated with more rapid progression to HIV disease and less. And we are looking at the SNPs right now. The sex, there doesn't seem to be any difference, but we do want to pay more careful attention to that. Particularly, the monkey studies generally use male monkeys. And so we're going to try to see if we can enroll or get some female monkeys into those studies as well to see whether there's something. So initially, in vitro, we can't see a clear correlation between response and non-response because we do have some female donors, but it's more complicated than that. Back there.
I think there is a hypothesis that the shocking, the kicking part is actually stochastic, so you can never eliminate all the latent reservoirs. And in this case, I feel like the kill part, we are quite confident, but the kick part, we don't really know what is the percentage of the latent reservoirs we can eliminate. And if we can only eliminate 50%, that may not be enough. We may have to eliminate 99%. But yeah, I don't really know that number, but how confident are we that in that aspect? If this strategy has to work, then I think you have to kick enough cells out of it.
Thank you for that question. You've clearly been reading the literature. Good for you. Good for you. That's a very, yeah. Your point is a very good one. The literature, and mainly work from Bob Silicano's lab, shows that you can come back even with very strong mitogenic stimulation, CD3, CD28. Not all cells become activated at a given time. You can come back later and activate cells that hadn't been activated the first time around. I think that's your point, right? That at a given time, not all cells, even with strong mitogenic stimulation, are not ready to receive that signal and begin to produce HIV particles. But the message from those studies does say that if you come back, there might be a time where those cells all become responsive. So we imagine these studies to take place over multiple years. It's not one shot, not one day. But I imagine that, the TLR7 agonist seems to be safe. We'll confirm this in humans every 2 weeks. So you can imagine going 10, 20 doses every other week. I'm hoping that at dose 20, we're going to pick up some cells that may not have been responsive at dose 1. And so hopefully with time, as the cells become responsive to the stimulus, hopefully we'll get it with time. If it was a race, I would say, I don't think we're going to succeed. But it's not a race. It's really, we can go slowly and gently until this reservoir is reduced. So I think the key is to have a pretty robust inducer and an inducer that reaches all cells at a given point in time. The 7 agonist, as you can imagine, that's what we're looking at. So for example, I told you there's a small proportion of CD4 cells that become activated. We've begun to dig deeper into that, right? It's important that some of those cells be central memory. If they're not central memory, then this doesn't make a whole lot of sense. And central memory cells are activated by this agent. So now what we need to do is intersect the two and ask, are the central memory cells that are activated also those that harbor HIV or SIV, and do they become producers? In other words, I'd like to connect these mechanistically. I think that CD4 activation is part of why these cells become producers, but I don't know that yet. But thanks for that question. Another question here.
Back to the monkey study. So I thought there were 7 other monkeys that you have not shown the data on. What happened to them? Because I'm really curious about the peach monkey, the one that had the laggiest response to it. I was wondering, what was their reset point? Because is there a correlation between the initial reservoir size and whether or not you can induce this virus back up?
Yeah, thank you for that. So those 7 monkeys are the placebos, and so.
I thought your study design was 6 placebos, 7 on treatment, and also 4 on treatment, so a total of 11 versus 6.
4 versus 7. 4 received drug, 7 placebo. Yeah, but your point's a very good one. We did look at the set point before ART, and we looked at the set point after ART. It is important to normalize to that. And when you do it that way, if you basically establish that relationship, the post or post-CART rebound set point, viral load set point, versus pre, that correlation still holds, that there's a half-log drop. But that's a very important normalization, because sometimes these animals, if you don't randomize them correctly, you may get surprised. So it did hold even if we did that rigorous analysis. So it wasn't that the lower set point animals were the responders, basically. Yeah. Other questions? I think there's been enough, David.
Thank you very much.